Assalamualaikum warahmatullahi wabarakatuh to everyone who is listening to this right now. My name is Nur Husna, representing Group PBL 17 and 18. For this forum, I will be talking about the ethical issues on gene therapy and stem cell therapy. Okay, so introduction. Ethical issues in genetic therapy involve the use of genetic science for human good and the avoidance of preventable harms. According to the Genetics Home reference under NIH, the questions regarding ethical issues on gene therapy include: how can good and bad uses of gene therapy be distinguished? Who decides which traits are normal and which constitute a disability or disorder? Will the high cost of gene therapy make it available only to the wealthy? Could the widespread use of gene therapy make society less acceptable of people who are different? And also, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Not only that, there are also immediate issues such as the control and confidentiality of results of genetic testing, possible discrimination against those found to have genetic diseases, allocation of beneficial genetic therapies. Less practical, but possibly more significant, dilemmas involve the use of genetic science in ways that go beyond prevention and cure of disease. First of all, CRISPR and its principles. CRISPR-Cas9 is a method of genome editing that exploits a natural DNA snipping enzyme in bacteria called Cas9, CRISPR-associated protein 9, to target and edit particular genes. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are segments of DNA of particular structure found widely in bacteria and archaea. In the wild, the CRISPR-Cas9 system is part of the prokaryotic immune system, which can snip out of the genome DNA acquired from foreign sources such as phages, which are bacterial viruses. The same molecular machinery is now being used to enable genetic material to be cut from and pasted into the genomes of other organisms, including eukaryotes such as humans. It might offer a tool for curing genetically based diseases. CRISPR and its mode of action. Formerly, CRISPR-Cas9 was adapted from natural genome editing process which happens in bacteria. The bacteria will capture a small part of DNA from the viruses which invade the bacterial cell. Then this captured particle of DNA will be used by the bacteria to generate DNA segments known as CRISPR arrays. The CRISPR arrays will cause bacteria to be able to identify and remember the same viruses and even the ones which are closely related. If the same virus attacks the bacteria will produce RNA segments from the CRISPR arrays to target the virus DNA. Later, the bacteria will use Cas9 protein or any enzyme to cut the DNA apart, which results in the death of the virus. The CRISPR-Cas9 system works similarly in the lab. Researchers create a small piece of RNA with a short guide sequence that attaches to a specific target sequence of DNA in the genome. The RNA also binds to the Cas9 enzyme. As in bacteria, the modified RNA is used to recognize the DNA sequence, and the Cas9 enzyme cuts the DNA at the targeted location. Although Cas9 is the enzyme that is used most often, other enzymes, for example CPF1, can also be used. Once the DNA is cut, researchers use the cell's own DNA repair machinery to add or delete pieces of genetic material or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. CRISPR's first human clinical trials, He-Jacques affair. Lulu and Nana are twin girls that were test subjects for genetic mutation. It has been claimed that the biological father of the twins is HIV positive. He describes targeting the CCR5 gene, which helps the HIV virus enter healthy human cells. The aim of this procedure was to make them resistant to HIV infections. He worked with 7 heterosexual couples in which the male partner was HIV positive and the women were HIV negative. After the couples produced embryos through IVF, he used CRISPR to cut the CCR5 gene, disabling it in hopes of making the embryos less vulnerable to HIV infection. He claims that of 22 embryos, 16 actually showed signs of successful CRISPR editing and 11 were implanted, resulting in a single pregnancy with twin girls who were born in November, which are Lulu and Nana. One twin, according to his test, showed signs that both copies of CCR5 gene it inherited were successfully altered, while the other twin showed that one version of the gene it inherited was altered. That so-called mosaicism, in which some but not all of the embryo cells are altered, is troubling since in this case it would mean that girl may not be entirely protected from HIV infection like a sister. The effects of this condition is unstudied. There is no independent confirmation of the claim nor has it been published in a journal. It's impossible to verify whether of the girls indeed showed successful CRISPR editing or not because the data have not been peer-reviewed and the fidelity of the gene editing process cannot be evaluated. Many scientists have strongly condemned the claim and called it human experimentation. A Chinese court has sentenced He Jiankui to 3 years in prison for illegal medical practice and handed down shorter sentences by 2 colleagues who assisted him. The court also fined He 3 million yuan. He was fired by his university in January 2019. Ethical issues surrounding the CRISPR baby scandal. First of all, not fulfilling an unmet medical need. In this method, the focus gene is blocked as it can be a doorway for the HIV virus. However, this CRISPR editing method is proven to be too extreme when there are already existing drugs for the treatment, in which they have been repeatedly tested in clinical trials. Number 2. The function for the new gene mutation is unclear. Alteration in genes can cause substantial change in how the gene works. The method had not been tested in lab animals yet, but had been hastily introduced into human patients. Problems with informed consent. Test subjects were not informed about the risks of off-target effects or about the facts that gene editing was a prohibited or ethically controversial technology. Number 4. Tests were operated secretly. The institution was not informed about the experiment, and the ethical approvals from hospitals were suspected to be forged for its own benefits. Next, the act was against global consensus for gene editing technology. Tests should only be done in clinical trials with meticulous and careful supervision only after a broad agreement by the public. The CRISPR babies were clearly produced under an unethical surveillance. Lastly, the perpetrator had not shown remorse on this turn of events. This can pose a problem where similar incidents might occur in the future. Other researchers might also conduct experiments under unilateral decisions. Introduction: Stem cell therapy principle. Stem cells have the ability to differentiate into specific cell types. The 2 defining characteristics of a stem cell are perpetual self-renewal and ability to differentiate into a specialized adult cell type. It can build every tissue in the human body, hence have great potential for future therapeutic uses in tissue regeneration and repair. So, how does stem cell therapy work? Stem cell therapy, also known as regenerative medicine, promotes the repair response of diseased, dysfunctional, or injured tissue using stem cells or their derivative. It is the next chapter in organ transplantation and uses cells instead of donor organs, which are limited in supply. Method: researchers grow stem cells in a lab. These stem cells are manipulated to specialize into specific types of cells, such as heart muscle cells, blood cells, or nerve cells. The specialized cells can then be implanted into a person. For example, if the person has heart disease, the cells could be injected into the heart muscle. The healthy transplanted heart muscle cells could then contribute to repairing defective heart muscle. Moving on to the ethical issues of stem cell therapy. Case 1. Moral. Many difficult questions engulf the morality of destroying embryos or using remnants of aborted fetuses to improve the medical well-being of other human beings. Proponents of embryonic stem cell research argue that during the natural reproductive process human eggs often fertilize but fail to implant in the uterus. While a fertilized egg has the potential to form human life, it is not equal to a human being until it has at least successfully implanted in a woman's uterus. IVF clinics often create more embryos than needed over the course of fertility treatments, and the excess frozen embryos are discarded. Case 2. Spinal cord injury. The FDA approved the first clinical trial of the HESC-based experimental intervention for spinal cord injury. This was enrolled a small number of patient subjects with recent serious spinal cord lesions. And anyway, HESC stands for Human Embryonic Stem Cell. Moving on, the trial's design and its ultimate discontinuation were controversial. Subjects were enrolled very soon after a serious injury, making understanding and consent challenging in this first-in-human trial, and in addition making it potentially difficult to distinguish between spontaneous recovery of function and remyelination attributable to the intervention. Patients with older lesions, though very probably in a better position to make decisions about trial participation, have scar tissue that makes remyelination unlikely or possible. The sponsor's premature discontinuation of the trial was problematic because data insufficiency renders worthless not only its own investment but also those made by patient subjects and investigations. The outcome had the potential to discourage pioneering stem cell research in the future. Case 3. Religious and moral conviction. The belief that an embryo is a person with the same moral status as an adult or a life-born child. Therefore, a person needs to be respected for their rights and interests. Taking a blastocyst and removing the inner cell mass to derive an embryonic stem cell line is tantamount to murder. With a different view of the moral status of the embryo, for example that the embryo becomes a person in a moral sense at a later stage of development than fertilization. Few people, however, believe that the embryo or blastocyst is just a clump of cells that can be used for research without restriction. Many hold a middle ground that the early embryo deserves special respect as a potential human being, that it is acceptable to use it for certain types of research. In 2003, the National Academy of Sciences established a committee to develop guidelines for institutions and investigators conducting HESC research. The NAS Guidelines for Human Embryonic Stem Cell Research 2010 comprehensively address permissible and impermissible categories of HESC research and recommend the establishment of embryonic stem cell research oversight committees to assist in the research review. I guess that's the end of the presentation. Here are the names of my group mates. That's all for me. Thank you very much. Stay at home.